Okay, let's go ahead and get started. We have Thanksgiving break coming, so there's nothing really due for the next couple days. You've got a lab, extra credit lab the week you get back. That lab will have a lab report associated with it, but it will be you'll write it in lab. So write lab, everything will be contained within lab for that. Your lab report, your current lab report, is due on Thursday after Thanksgiving break. All right, I feel like I'm missing something. Am I missing anything? I can't think of it though.
We have an exam the week after. It's on
The
Friday.
exam like last day of class, right?
Yeah.
Okay.
It's December 5th.
Exam last day of class. Okay. That's at 2 to 5. Is that what our schedule? We've got the very last exam block there is.
Oh yeah, it's 2 to 5 on
Yep.
12.
Yep. So if you're one of those people, excuse me, that needed to take it early, I know a couple people have talked to me. Maybe we should start coordinating that. Not right now, but talk to me, and I'd like to try and coordinate that to either get you in somebody else's class or have everybody take it together. Okay, so let's go on. We were questions on things we're covering. Okay, so then let's go on, and we were talking about carbohydrates last lecture, and what I want to do is, what I didn't emphasize is the transition, or how you look at a Fisher projection and turn it into a Haworth projection, and how you take that and put that into a chair. So let's do that. So talking about Fisher projections, Haworth projections. Is that not how? Is it just go ahead and capitalize it? I think, yeah. I looked it up before I came in. And chair projections. Okay. And so let's say we have glucose. Let's see if I can do glucose in the linear form. We'll go this way. So, right, glucose. So all of our 6-carbon sugars are going to look like this. And of course, for a carbohydrate, for a monosaccharide, you can have less than 6 carbons, right? So we talk about glycogen, not glycogen. We talk about glycerol. Glycerol is a 3-carbon carbohydrate, right? Glycerol looks like this. And that's been important if we're going to synthesize. It's an important part of our membrane lipids and also our triacylglycerols. But that's a 3-carbon carbohydrate. So it's not that they're all 6 carbons. There are 5-carbon things that are important as well. But we are just covering these 6-carbon ones in terms of this class, in terms of detail at least. All right, so glucose. This was on this side. Was it this? Bridget, did I get it right?
No.
Okay.
It's because glucose gives you the middle finger, and so it's the second one down.
It's this one that flips?
Uh-huh.
Okay. Okay, so that's our Fisher projection. And so then if you want to turn that into a Haworth projection, the way you do that is you're just going to turn this thing on its side. All right, so if we turn this on its side, then you have this. All right, and so now what I can do is I can draw my Haworth projection. And the first carbon, okay, so if we think about the numbering here, once again, 1, 2, 3, 4, 5, 6, and 1, 2, 3, 4, 5, and 6 will be up here, or it would be down if if this was not a D form of the sugar. But so in terms of the Fisher projection, what do I do with the beta alpha anomer?
Beta goes up.
Beta goes up, alpha goes down, but I don't know which is which off of the Fisher projection. It would have to be specified, right? So yeah, we could at this point we just go a squiggle unless I said alpha anomer, beta anomer, alpha or beta, whatever glucose. All right, so we've got then two is down. You see this in terms of the tipped over. This goes down. This one goes up. This one goes down. 4 is down. 5 is this oxygen. Okay, so 5 goes down, but 5 is this oxygen. And then if we have the D form, that's going to put this OH up here. All right, so this would be D-glucose. And we're just going to go with that and talk about the cyclic form. It would be D-glucopyranose, but I'm not going to bother with that nomenclature. Questions so far? All right, so let's go ahead and let's say I was asking for, just because we're about to do the chair formation, let's say this was, which one is this? Which anomer did I just draw?
Alpha.
The alpha anomer. So this would be alpha D-glucose now. And we want to go to the chair form from that. You guys had Dr. Dan for organic?
Yeah.
He's so careful with his chairs. Maybe they all are. I sat in on that lecture. I'm not so careful with my chairs. There, that's a chair. So you do your chair form, right? You would draw like that, and then in terms of what I have up above here, I've got my points on my chair, right? I've got a point there, and I've got a point there. And what we think about off the chair is we're going to have axial positions and equatorial positions. So my axial positions go, you take them and you stick them up from the points, and then you alternate going around. Okay, those are my axial positions. And then my equatorial positions don't go up and down. All right, I'm not going to be more picky than don't. So as long as they're not up and down, they're clearly not up and down, I'm good with it. And so now I can look at where my OHs go. And if I look at 1, it's definitely down, right? The 1 position here is definitely down. And so I'm going to put an OH there. My 2 position is definitely down, which is definitely not up, right? So it goes there. My 3 position, let me go ahead and number these again. 1, 2, 3, 4, 5, and then 6 is going to be out here. My 3 position is definitely up, right, which is not down. My 4 position is definitely down, which is not up. And then once again, we're doing D-glucose, so this is gonna come out. That's an angle there, like that. Okay, and then I need to go back and definitely, today's a really definitely day. Definitely is the word for today. But I need to go back and put H's on if I've got these little lines drawn, because otherwise they're methyl groups. Questions on that? Yes?
When you're drawing like the axial, how do you remember which direction it goes?
How do I know which one it goes?
Like right.
That's a good question. So let's go ahead and draw a beautiful chair again. No, that's not beautiful enough. Okay, there's my chair. So down, right, where the way the ends are pointing, and up. Okay, and then alternate. So that this one right here went up, so this one goes down. This one went up, this one goes down. So they're alternating, up, down, up, down, up, down. This one's down, so this one's up. And then I've got all of them.
But then, like, how about when you do
The equatorials?
or, oh,
The equatorials are not up and down.
how do you know, like, right, left?
Don't worry about it. I'll accept it regardless.
Thank you.
As long as it's not up, as long as if you did this, I won't accept it,
Right.
right? I need to look at this and I need to see one that's clearly up or down, and then one that is clearly not up or down. That's all I care about. All right. That would not cut it in organic, but it doesn't here. Other questions? Okay. All right, then let's go back and talk about some of our polymers here. So we were talking about, actually it's coming up on Thanksgiving, so let's talk about gravy. So when we talk about plants, right, plants have a carbohydrate polymer they have, which is a mixture of amylose and amylopectin. So they are the glucose storage polymers for plants. And so when you think about, well, kind of any flour you have or whatever, they will have different amounts of amylose and amylopectin. Let's talk about the structures of those. So amylose is a straight chain polymer, an unbranched polymer, let's say, a unbranched polymer of glucose with, I'm going to write alpha rather than play around with trying to get the right thing, but alpha D, or not alpha D, alpha 1 for glycosidic bonds. All right, so what does that look like? Well, with that sort of designation, you should be able to draw it, or you should be able to say that. So let's say that I would have a series of glucose molecules. I'm going to draw like this. And for testing these, you will have to be able to draw them. And I'll say, like, draw me 6 residues of this polymer showing branches if it's branched. This one isn't, right? I haven't drawn 6 here, but that's generally what I do because if you're going to do branches, you can't do branches with 4 very well. And then you have to show that it's glucose. So in terms of grading these, I would give, points for having the 1,6 glycosidic bonds. I would have points for having it be glucose. If it was branched, I'm sorry, 1,4 glycosidic bonds. I'm thinking about our branches. You'll see those in a minute. But this is amylose.
So yes. Is it based on how many are in the polymer that makes it like glycogen versus amylose versus amylopectin?
Well, it's based on the branching.
Okay.
So glycogen branches, amylopectin branches. Glycogen and amylopectin branch a little bit differently, although you wouldn't draw the difference because you'd have to draw too many residues, and that would be your whole quiz or test. Amylopectin is a branched polymer of glucose. Okay, the main chains are made up of glucose residues with alpha 1,4 glycosidic bonds. Okay, branches occur every 24 to 30. Forgot to get my notes out, but they occur every 24 to 30. And the branching is an alpha 1,6 glycosidic bond. Say, and use alpha 1,6 glycosidic bonds. Okay? And so if I was to draw 6 residues of this, let's see, I might be lazy. If I can get these, I'm going to be lazy. Magic. Okay, so let's say I've got these 4 residues. That's my main chain, and then I'm going to have 1,6 glycosidic bonds coming off of this for branching. So imagine we have a branch right here. That's going to come up. That's the 6 carbon that's off of, right? So it's 1,6. So there's another glucose up here. And it's going to go on, and we're going to have a chain there. Should have left myself some more room. You're probably not there yet drawing, right? I'll put my branch here so I have some room on my page to actually draw a few more glucose residues. All right, and then it just keeps going. All right, so we have this polymer. It's just like amylose, right? And then every once in a while, every 24 to 30 residues, we're going to have a 1,6 branch that's going to start, and then it will just continue. And so if you were to cartoonify this, you'd have something go, you'd have a polymer going, it would branch, that branch would branch, the branches would branch, more branches and more branches, okay? And really what it does is in three-dimensional space, it's not going to look like that. The way it's going to look is you're going to end up with, I've got this thing going and it branches and that branches and that branches and that branches, and all the branches have branches. And essentially, a starch molecule, actually a glycogen molecule has 50,000 glucoses in it on average. I actually don't know. For amylose, and the ratio of amylose to amylopectin is different depending on what plant starch you're looking at. So cooking-wise, you care about how much amylose is in something as opposed to amylopectin by how well it can thicken things up. And so you will pick your starches depending on what you want to happen with that. So let's talk about Thanksgiving. All right, so we've got Thanksgiving, gravy. So how do you make gravy? I make a vegetarian gravy, but let's say you were doing it with turkey. What happens there? Oh my gosh, you guys need to help out in the kitchen more.
I don't like gravy.
Put some flour in a pan.
Yeah.
Add some like fat, grease. You kind of stir it. It's kind of like making a roux.
Yeah, it's exactly like making a roux. Very good. What you do is you put grease in with flour, right? You're coating it, and you're trying to separate out all the starch granules, essentially. But the making the roux bit is you put the grease on, you try and get all that flour all those little starch granules separate from each other, and then you're going to add some water, and it's going to swell it up. So what you have is a starch particle, or a starch granule. Okay, it's going to look like this, where the blue is amylopectin, and let's say the green here is going to be amylose, kind of woven through it. All right, and what you do is you add water to this. I mean, you add gravy, you add fat to get them all separated so that the water can get at them. And you add water at them, and water and heat, and these separate out. And so what happens is your amylose particles get separated, and they get some water in them, and they swell up. So let's say swells with water. And so from the perspective of thickening this stuff up, what's happening is I've now got these big starch molecules, right? I got these big amylopectin molecules that are swollen up with water, and they're trying to move past each other in solution. And they're big, and so they have trouble doing that. And so that slows things down. But in addition to that, you actually get a gelling, just like the gels we made in lab, or just like Jell-O, really, in that your amylose molecules are going to get out here, and they're going to get they get released, and then they go ahead and tangle, and they trap water between them. And you get more of your thickening power from this. Okay, so you end up with trapped water, can't move. I'm going to write gel. Let's say freely. Okay? And so, gosh, I should have looked it up because I've forgotten at this point. But I think rice flour has very little amylopectin in it, and it's mostly amylose when you think about this. And so rice flour is better at thickening things than, say, the regular wheat flour that you might use. You have to like the taste of rice flour, though. Questions?
So amylose traps the water?
They both trap water. But amylose, what's going to happen with the amylose, so you get the thickening by these separating out. The amylose is going to actually swell up and get bigger, so you'll get some thickening from that, but you get more of the thickening by these strands of the amylose. Did I just say amylose twice? Amylopectin, as I was just talking about. The amylose, they all go and they tangle up, and that's a gel, just like the gels we used in lab, just like Jell-O, where water is trapped by polymers around it and it can't move very well. So you have to see that if you look at cooking videos. So I've been trouble sleeping last night, and there was what was it? It was turkey stuffing, whatever. I'm vegetarian, so I'll have to get some, Impossible sausage, but we're watching Chef John. Does anybody watch Chef John? Oh, okay, well, he's funny. But I think I want to try that. Okay, so that's how gravy works, okay? And that's also the structure of a starch granule, right? More than that, know the structure of a starch granule, an amylose and amylopectin. For humans, we don't have, or animals don't have two separate polymers. Animals just have glycogen. And I'm going to say for glucose storage. Okay, and what animals are doing in terms of glycogen for glucose, or for the structure of glycogen, glycogen is a branched polymer like amylose, but it branches more often. So it's a it's a polymer of glucose. with 1,4 glycosidic linkages and 1,6 branches. Every 12-ish, let's say, 12 residues. So if you had to draw a picture of a structure for glycogen, essentially it would look the same as the structure for amylose up above. Look at that. Oh wait, I have to draw part of it still. Oh well, I can do that. All right, so you could draw the same thing because I'm not going to have you draw, 90 glucose residues in a polymer. Okay, so that I can see that you actually did it every 24 to 30 as opposed to every 12. But so once again, here's a 1,6 branch. And then once again, when I'm grading these, like if I was grading this, I would have it be like, I don't know, 8-point problem or something. Did you make it out of glucose? So do you have the stereochemistry rate for those? Do you have a 1,6 branch? Do you have the 1,4 linkages? Did you get the linkages correctly? Yes, Jacob.
Since that structure is so similar, why is there a difference in sometimes the branch?
What I don't know. Natural selection is the answer, but I don't know what the advantages. What the advantages of having a branched polymer is you have more ends. So let's talk about that real quick, because the ends, it's going to operate when you think about enzymes building this. They're building it this way. I'm not really showing it here, but for glycogen, there's the very end. Let's say this was the very end as opposed to a branch coming off of a branch to get to here. This was the end. You actually have a protein that it starts on. It starts in a protein, it's linked to the protein. That protein kind of serves as a starting point, essentially. And then it branch. So if you looked at the middle of a glycogen molecule, there'd be a protein in the middle that was just hooked to. But there are directions in these, okay? So we can talk about two ends. So just like in proteins, we have an amine end, a carboxylic acid end. We have a 5' end and a 3' end when we're talking about nucleic acids, right? We don't really have polymers for lipids, so we don't have ends there as much. But what we have here is we have a reducing end. And when I say reducing end, I mean this residue. Okay, and we have non-reducing ends for the polymers. Okay, so you want to think about that. If we were to scoot up and look at not this one because that's the same thing, this is the reducing end, so it's the 1 end. But consider it the residue is at the reducing end and the non-reducing end. What's the difference? The difference is in reactivity. So if we think about, let me come down to this one again. So if we think about where these come from, right? They come from aldehydes. Was it this for glucose? I'd have to hold on. All I need to do is tip it back up. Yes, it is. Okay, there. So an aldehyde. If you take this aldehyde and you put it into a tube with some, silver's a great, you put it in with some silver +1, okay? And what it'll do is the silver will react with it, and we'll get a carboxylic acid over here instead. This is, I think Tollens. It's the Tollens test. I might not have that exactly right. But this is a test that you used to do in organic chemistry to see whether you had whether you made an aldehyde or not in terms of reaction. Now you just do NMR for everything.
It is called Tolan.
Okay, thank you. Show it up on your MCAT.
Oh, no, I went back to our organic because we did talk about this. I just
Okay.
messed it up. Yeah, we did.
Do you guys do that in lab? Or you just talked about it?
We just talked about it.
Yeah, I had to do that in lab. It's cool. It makes a mirror on. So essentially, what happens is it's reducing, right? So the sugar what happened to the sugar? It got oxidized. So what's actually happening with our silver is our silver is going to go ahead and turn to silver solid, and it's going to plate out around your test tube, and so you end up with a silver a mirror essentially on your test tube. Yeah.
Wait, but those ends look the same. I don't really understand how you guys.
So it's the aldehyde that's reacting, right?
Mm-hmm.
The aldehyde is at the 1 position. And so I'm giving you the residue. So for this, the way it's written, right? This isn't the actual reducing end here. It would only be the reducing end if I did the official reducing end. But in terms of talking about, if we're looking at a stretch of polymer, right?
Mm-hmm.
This is the reducing side. This is the non-reducing side. Okay, but this, if we look, so it's the 1 is the aldehyde, right? So we have a free aldehyde here. We don't, this is a heavy acetal, right? The reactivity of this carbon has 2 oxygens on it. That came from an aldehyde. It's different than the reactivity of all the rest of those. This reacts, so this can open up and react and give you that silver mirror. It can reduce other things. The other one, for these other ones, it can't because it's tied up. So here I've freed it up, right? For these over here, it's tied up. It's attached to something else. So you can't open that back up, if that makes sense. And so the reactivity is where the terminology comes from. Does that make sense? So it's the 1, it's the thing that was the aldehyde, but in regards to this, we're just talking about this end, okay? And why I'm emphasizing the residue is sometimes I have people tell me that's a non-reducing end and that's a non-reducing end. They label all the hydroxides. This is all hydroxyl groups. This is about telling a direction on a polymer, just like it's about telling a direction on a polymer for proteins, and just like it is for nucleic acids. So we talk about the 5' end and the 3' end. Nobody cares that you're saying it's that phosphate. They care that residue, that's the end of the polymer. Does that make sense? So it's about a polymer direction. And so where this is useful in nature, though, is if you think about a cell's trying to store this stuff and trying to store it fast, that if I, once again, don't draw it as a sphere, even though that's the way it would look, I go out like this, so I'm growing, right? We've got some protein here that it's nucleating from. And if it branches, now I have 2 places it can grow from. So it can make it twice as fast. So now it can 3 times as fast, and 4 times as fast, and 5 times as fast, and 6 times as fast, right? Because I have 6 ends that I can add to make this polymer bigger. So that's the advantage of having ends. I can't say why glycogen has more branches than amylopectin. And then if you need to release, so we've got a glycogen, let's say this is our glycogen molecule in your liver, you're between meals, you're running from a bear suddenly, and you need glycogen release. You need it broken down fast. Well, now you have a bunch of ends, right, to break this down. So instead of having a single linear polymer like that needs to be broken down to release glucose from glycogen. This you'd be able to do 1, but here 1, 2, 3, 4, 5, 6. I can do this, break this down 6 times as fast.
But I feel like what type of end would be there?
What type of, these are the non-reducing ends. And the reducing end is over there stuck onto the protein. Okay? Does that make sense? Okay. So what was that? So let's go on and talk about cellulose. There's a picture of cellulose right there. But I'm going to describe cellulose. So cellulose is a structural polymer, and it's different in that it has a different linkage. Not very different, but different. So cellulose is a polymer of glucose. So just plain old glucose again. Glucose residues linked together by beta 1,4 glycosidic linkages. Okay, it's unbranched. All right, most animals can't digest cellulose because they don't have enzymes to break down 1,4 glycosidic linkages. And so if we were to draw this is what I talked about the other day. What do you want, squirrely bonds or upside downs?
Staggered.
Staggered. Staggered going up. I'm not going to do staggered. It's a great idea, though. All right, so we're going to do upside down.
I thought we were doing squirrel.
You can do it however you want.
I'm gonna do it staggered. I hate the upside down. That looks like an abomination.
I asked and nobody,
I gave you an answer.
all I got was staggered and louder. But you can draw it how any of the correct ways to draw it, okay? But you need to be able to recognize all these other, oops, that's not right. Oops, that's wrong. Like that, okay? And so what we've got is it's a beta 1,4. The beta is here, right? Each of these, if we just look at the up ones, each of this is going up. It's going up. This is going up, but that's upside down. It's going up. Okay? And then it's gonna connect to the other one over here by coming up from the bottom because this is glucose. Glucose, this hydroxyl has to be down. Questions? Okay? So just straight, that's how you can draw them like that. And that's kind of how they exist. So this is an example of just in 3 dimensions what this would look like. And so these tend to, you get nice hydrogen bonding between the residues. And so these tend to be pretty straight chain in solution. Just kind of go straight. Doesn't mean they can't bend around. Questions? Okay, so chitin is a variation on cellulose. Chitin, like that. So chitin is, this shell material, okay? Insect shells. What else does it go into? Crustaceans. And so what this is this is it's a glucose, but it's actually, it's an N-acetylglucose. Okay, so this is a polymer. You don't need to know this one, so I guess I won't write it out besides just saying that chitin is going to be one of these straight chain polymers again. It's structural, right? And it's made up of glucose residues, except the difference between it and cellulose is that we have these N-acetyl groups on them. So down here where you'd have a hydroxyl, you've got an N and then an acetyl attached to it. So what happens in nature is glucose is kind of the base molecule used for almost all of these, and then it gets modified in various ways. It's not always that, but a lot of the time. Sophia.
I'm just wondering, like, how does, like, you know, like, how this makes it different from cellulose, like, the...
Oh, what
like,
chemically?
Harder, like, yeah, like, what is happening to make it, like...
I don't know. I don't know in terms of physical properties. And I don't know whether this gets cross-linked, right? Doesn't Dr. Pesh does work with lignin? Doesn't lignin go in, and does lignin just mix in with the cellulose, or is it actually chemically cross-linking with it? It's cross-linking. So you have other molecules in cellulose that are cross-linking, and so there may be things that are cross-linking here as well. We'll talk about the bacterial cell wall, and I do know how that gets cross-linked. Okay, questions? All right, I think we're ready. Oh, we are ready for the bacterial cell wall. Okay, so we can talk about many bacteria have a part of their cell wall that's made out of peptidoglycan, which is a mixture of a carbohydrate polymer, it's a polysaccharide, and protein crosslinks. So the peptidoglycan layer in bacteria. And the peptidoglycan layer, they're cartooning it with this. Okay, so you got your bacteria here, and then we've got in the layer outside our membrane, we've got a bunch of polymer chains, and they're cross-linked. Okay, we'll look at the actual structure in a second here, but not quite yet. So the peptidoglycan layer in bacteria is made up of polysaccharide chains cross-linked by peptides. And so if we want to look at what that's what that structure looks like large. And if we want to look at what the actual molecular structure is, it's going to look like this. I'll make this bigger. And so I don't expect you to know what the structure is here outside of generally it is a set of, we've got a set of, this is actually disaccharides. So rather than have the same residue over and over again, what you've got is you've got alternating residues. Okay, so we've got N-acetylglucosamine, all right? We just saw that up above, right, in the chitin. And then we have N-acetylmuramic acid, which is also glucose, but we made an acid out of one of those groups. Okay, so it's these are modified glucose molecules or monosaccharides that are polymerized together in these chains, and then they get cross-linked by amino acids. Okay, and it actually does show you by color there. It's a glycine cross-link. Yes.
Can you tell what cross-linking means?
Cross-linking, good question. So cross-linking, what it means is that if you think about this, we've got these three chains here. Here, let me use like these guys. Okay. Oh, maybe not. The pen's attached to them. Okay, so you could think about these being your chains, although they'd be more flexible than this. Okay, you've got them running side by side the bacteria, but they're not very structurally alone. They're not very good because they can come apart. They're also going to be wiggly as well. So the cross links are, we've got a peptide that goes from here to here, and then one that goes from here to here. So it's like our disulfide bonds, but longer. Okay. Does that make sense? Yeah.
And they go to the same residue, like of glucose.
Yes, they're going to this, it looks like they're linked to the same residue across here. Okay, but you get a lot of strength from adding these peptide cross-links across here.
So like, you want us to know that they cross-link and that they create strength, and that's like the point.
Well, I'm going to talk about antibiotics and penicillin and things like that. And that I want you to know the penicillin story. I want you to know that it's a protein cross-link. But I don't care that you know that it's N-acetylglucosamine and uramic acid. I don't care that you know that it's that there's these glycines here. I do care that you know that it's DD transpeptidase is the enzyme we're inhibiting with penicillin. Yes.
So, wait, what's the thing happens with like the glucose things like, sorry, I'm just trying to put it in
Yeah,
perspective.
so the bacteria, when it's making its cell wall, it's constantly kind of remodeling. It's tearing down its cell wall and it's rebuilding it because it needs this is a constant process here. So it's tearing it down and remodeling it, and whenever it's makes some more cell wall, it makes these chains, and then there's an enzyme that goes ahead and adds these peptides to hold these chains together. All right, so the protein cross-links are added by an enzyme called DD transpeptidase.
Always or in bacteria?
It's just that you only have a peptidoglycan in bacteria, as far as I know.
Cool.
So the protein cross-links are synthesized by an enzyme called transpeptidase. I'm going to say DD transpeptidase. If you just said transpeptidase, that'd be okay. Okay. Penicillin and the other cillins inhibit this enzyme. They get wrong with penicillin. So when you've gotten a cillin, what has it been?
Amoxicillin.
Yeah, amoxicillin is the one that's really used in humans.
Yeah.
All right, and so when we look at this, we want to look at structures here. This is penicillin. I'm just going to draw some arrows. And then this is amoxicillin. And what do they have in common?
That weird S nitrogen circle thing.
Right, right. And the big thing is beta lactam. Beta lactam is what it's called. We got the square, right? The four-membered ring. How does it what?
Isn't that super unstable?
You know, it's not super unstable. There is an issue that, you know, my research used to be doing HPLC and validating pharmaceuticals from middle to low income countries. And amoxicillin goes bad worse than a lot of things. So if you let water get at this, so you get your amoxicillin tablets and you want to keep your Z-Pack, or you get them and they're packaged in a, what are the packs called again? I can't remember, but you know, you've got a package in the plastic with the foil on the back. When they're in there, that's great, but then what happens in a lot of lower to middle income countries, they break those open and they resell them and hand them out to people in bags, and then water's able to get in and it degrades faster. So we've tested things, and if it's still in that original packaging, usually rock solid, it's just fine. And then if it's gotten moved out of that packaging, you have problems. Heat and water, but water is a real problem. Okay, so that's what you can get amoxicillin, but if your physician is worried that you might have resistance, the bacteria then developed resistance to these, and their resistance, I think I didn't write down the enzyme. I didn't. That's too bad. But essentially what we've got is we have, these are going to, I've kind of made a mess of this. Hold on. So these guys inhibit the transpeptidase, but what bacteria will do is, I've forgotten the name of the enzyme, it's a beta-lactamase, I think, actually. It's a beta-lactamase. They synthesize a beta-lactamase that goes out and actually, so they're just actively to fight this, they synthesize an enzyme that breaks down the penicillins. Okay. So resistant bacteria, why I've got bacteria like that. Resistant bacteria synthesize beta-lactamases to break down cillins. Okay, so this is a resistance, this is a method how bacteria can become resistant. And all this is actually happening outside the bacteria. So these crosslinks get made outside the cell, right? They're in the wall. And then these beta-lactamases will get exported out there as well to get rid of the cillins. And so that if your physician's worried that you have something that's resistant, rather than just give you amoxicillin, they'll also give you something called clavulanic acid. Okay. So clavulanic acid can be co-administered with the amoxicillin to keep it effective against resistant bacteria. And this is clavulanic acid right down here. Let me see if...
Why don't bacteria grow resistance towards that?
Why don't they get a resistance against that? They probably do. I mean, so you've got multi-drug resistant bacteria. There are various things that you would give people. So you don't give amoxicillin for every bacterial infection. We're going to talk about it. When do we talk about that? I think it's a second semester topic, but you can talk about quinolones. They're not quinolone. Now I'm blanking on the name, but there are specific drugs they'll give you in the hospital that are different than things you'll get prescribed outside of the hospital. So they're looking to take care of. It's more broad spectrum. One of the problems with those is that anybody heard of C. difficile? C. diff, yeah. So C. diff infections. So it's a really kind of virulent bacteria that 5% of us just have. We just have it going around, and your usual intestinal fauna keep it in check. So the bacteria that are in my gut right now, if I have C. diff right now, I don't know whether I do or not, but if I had C. diff right now, the fact that I have a whole bunch of bacteria in my stomach, they're out competing and they're keeping my C. diff population low. If I go into the hospital and you kill off all the bacteria in my system, I can get a C. diff infection, because what happens is that if C. diff is resistant, fluoroquinolones is what I want to say. But you can they come roaring back because they're left, and now there's nobody competing with them. So that's where the C, you hear about people getting C. diff infections in the hospital. They've been given antibiotics, everything else has been killed off, and they had a resistant form of C. difficile. But anyway, so I don't need you to know that story, but I do want you to know this story, okay? So know about that the penicillins are inhibiting the transpeptidase, which is important for the protein cross-linking of the peptidoglycan layer. And for the structure of the peptidoglycan layer, you just need to know that it's polysaccharides cross-linked by proteins, or by peptides, we might say, is more correctly. And then amoxicillin, penicillin inhibits that. And then I would like you to know that, that bacteria can be, because it's an interesting mechanism, but it gets used in other forms as well, that bacteria can produce these enzymes, beta-lactamases, that will break down the cillins, and then you can co-administer something to stop them. You notice anything interesting about the structures here of the cillins and the clavulanic acid? Okay, well if we look here, yeah, there are different things they're saying, but we've got a ring here that doesn't have a sulfur, right, it has an oxygen. But we do have a similar shape. There's a lot of similarity here between the shape here and there. And these are a substrate for beta-lactamases. This is an inhibitor, right? It looks kind of like those things. And if it would be the best inhibitor if it looked like what? The transition, whatever the transition state is for that reaction. Questions? All right, let me see okay, well, I think we'll stop there. I'm not going to get into glycosaminoglycans until Monday. You guys have a good weekend. See you Monday.